It is my great pleasure to introduce our PI speaker for today, Dr. Sylvia Erhardt. Sylvia did her PhD at the University of Cambridge with Dr. Azim Surani and her postdoc at the University of Berkeley with Dr. Gary Carpin. She started her own group in 2008 at the ZMBH in Heidelberg University, where she was until pretty recently, so about a year ago, her group moved to KIT. Sylvia and her group have published many exciting studies on the epigenetic mechanisms of centromere identity, segregation, and cell cycle progression, using both Drosophila as well as human cells as models. So we are very excited that you could be here today, Sylvia, and we're very excited to hear your most recent work.
Thank you so much, Parhuta, for this introduction, and thank you everybody from the Fragile Nucleosome team for the invitation and the great work that you are putting out in this difficult times that hopefully come to an end pretty soon. So I'm going to talk about centromeric chromatin as usual and the balancing act that maintain genome stability. So it's sort of a little bit my generic title, and I will start with data that is already published, some of it for quite some years, but I want to show you those data as an introduction for data that is still unpublished, and we are hoping to publish pretty soon. So now I can move my slides. So I don't really have to introduce that here. So canonical nucleosomes that you can find throughout the genome on a metaphase chromosome. What I really want to introduce to you here is CENP-A. CENP-A is a histone H3 variant that replaces a subset of nucleosomes at the primary constriction of a metaphase chromosome, the centromere. And so CENP-A is the base on the chromosomes for the site where the kinetochore assembles during mitosis. So here that's the Drosophila kinetochore that you can see here. And so the kinetochore is then required during mitosis and meiosis to recruit mitotic mitosis spindle, the microtubules, to then have accurate chromosome segregation. And importantly, the centromeric DNA that is underlying the centromere here is not that important. I mean, it is important, but it doesn't define where CENP-A and the centromere is localizing. So CENP-A is an epigenetic determinant of the centromeric region. And so I just want to show you one quick movie. It's a very old movie, but I love it. That is a partial depletion of CENP-A in Drosophila cells. And you can see in green, the mitotic chromosomes just don't know where to go, how to bind properly to the mitotic spindle. So just to show you how essential really this histone variant is. And so because the CENP-A nucleosome and the CENP-A histone is so important, many labs have worked on how CENP-A finds the centromere and how CENP-A gets loaded. And what I show you here is just what we know from in Drosophila, our model system. There we know that like every good histone variant, it is replication independently loaded to chromatin. In case of Drosophila CENP-A, also called CIT, it is loaded during mitosis, depending a little bit whether you look at cells or embryos or other tissues, somewhere in mitosis. And so you need a priming of Cal1. Cal1 is the loading factor. It's the functional ortholog of HCHERP. You may have heard it's the mammalian loading factor of the mammalian CENP-A. And so during prophase, you then have Cal1 load CENP-A to centromeric chromatin, which also requires an inner kinetochore protein called CEMP-C. There is a monoubiquitination of CENP-A that is Cal1 dependent to stabilize the complex to load it to centromeric chromatin. And we recently also showed that that whole loading process is coupled to mitosis duration through a direct interaction with the spindle assembly checkpoint protein and RCC complex component ZW10. So the checkpoint has to be satisfied and CENP-A loading has to be completed to really have mitotic duration and mitotic progression going on. However, we know from many different cases that CENP-A mislocalization can occur. One very well studied example is actually different cancers here. That's a prostate section from a prostate cancer patient, where you see in red that there's massive overexpression of CENP-A or too much CENP-A. Also breast cancer and other cancer types have a misexpression of CENP-A. And many years ago, when I was a postdoc in Gary Carpin's lab, Patrick Hoyne and myself, we did a study where we showed that when we overexpressed in cells or in flies CENP-A, here CENP-A GFP, for instance, we get ectopic incorporation, ectopic kinetic formation, and chromosome segregation defects in those tissues, indicating really that CENP-A is required and its loading is required for accurate chromosome segregation. So what do we know about loading? How does it work? Many questions. I showed you with CAL1, the loading factor. However, we had other questions. Is the loading when you have too much or when you have a cancer cell, is it the same? So we looked at the CENP-A protein here in flies. It has a very long N-terminal tail here in turquoise. And there's more known about the histone fold domain and what it does. Also for CENP-A, has a CATD domain, which is required for centromeric loading. So not much was known about the tail, so we just decided to chop off in 25 amino acid increments the tail and see what happens in terms of ectopic loading, in terms of centromeric loading, etc. And what we found was kind of at first a little bit confusing. Basically, we could chop off everything of the tail and nothing much happened. We overexpressed that it goes into the nucleus, it binds to chromatin, not much happened. Protein amount was about the same. However, when we depleted the last 25 amino acids, what we did see is that CENP-A wasn't there anymore, a few little spots. And when we depleted the proteasome, we saw that it's actually stuck in the cytoplasm. So what we found is that within that region, it actually has a bipartite nuclear localization sequence. And when we depleted that bipartite nuclear localization sequence, or just those 3 arginines, what we found is that CENP-A, which we then call B3, behaves just like it doesn't have a tail. So what we saw here really was just an effect of the protein not being able to go into the nucleus, and therefore we see that massive defect. However, we used those mutants that we had, the B3 or the delta N, where the entire tail was gone, to screen factors that are known to interact with CENP-A, well, not only at the chromatin, but also in the cytoplasm, for instance, or for loading. And here, CAL1, that's a loading factor that I introduced earlier to you. You can see if you co-express one of the mutants with CAL1, we get a loading of this nuclear localization deficient protein to centromeres, and only to the centromeres. And just to remind you, if you overexpress massively CENP-A, usually you get it along the chromosome arms. And that is something that we saw when we co-overexpressed RBAB48, or CUF1 also known. And interestingly, what we saw here is that the protein also only goes to the arms. And here, marked with CEMC at the centromere, you don't see it. So RBAB48 really seems to be involved in ectopic CENP-A loading. And since RBAB48 is a protein that is part of many protein complexes, we did mass spec analysis, and to cut a published story short, we found that the NURF complex is really responsible for loading CENP-A ectopically along the arms. We found almost all the subunits of the NURF chromatin remodeling complex to be associated with CENP-A. And so our conclusion, our model really was that you need CAL1, the loading factor, to load CENP-A specifically to centromeres. So there's also the specificity that comes with CAL1. If you have misexpressed CENP-A, you need the NURF complex, and there are more details in there that I didn't talk about it right now, to actually load CENP-A ectopically to sites along the chromosome arms. And quite interestingly, we also then did a study where we used the International Cancer Genome Consortium and the XENA tool to see which proteins co-misexpress in different cancers. And you can see here, those are many different, see that is 500 samples, different cancer patient samples where CENP-A is highly misexpressed. And then we looked for co-misexpression of other chromatin remodeling factors. And we found, for instance, here, we found that the NERD complex components are co-misexpressed with CENP-A. So indicating that perhaps even in human cells, the NERD complex may be involved in ectopic CENP-A localization. The Al-Musni lab has published that the DAX complex is also involved, so there might be also a crosstalk of those two different things out here. So that was all with misexpressed protein. And then we realized we have a fly mutant that we worked on for quite some time, where CENP-A is actually also misexpressed. And that mutant or that protein is called CRAC-14. And you see here CRAC-14 embryos, CRAC-14 mutant embryos, and in red is CENP-A. And you see they massively overexpress CENP-A and localize it to chromatin. We also then did mass spec analysis, and we found that CRAC-14 and CENP-A do interact with each other. We also see that they directly interact with each other. And so we now really wanted to understand how is this CRAC-14 involved in CENP-A loading, and what is the function of that? And so that study has been started by Veena Matthew, a previous PhD student in the lab who has left quite a few years ago, and it has been taken up by Sarah Doppler, and Sarah is also almost about to finish her PhD now. So what we know about CRAC-14 is that it is also part of several different larger complex. The name comes really from the CRAC, the chromatin accessibility complex, where it interacts with ACF1, ISY, CRAC-16. Those two proteins are histone fold domain proteins themselves. However, CRAC-14 can also be found in the attack complex or in the DNA polymerase alpha complex, where you have it here, the leading strand at the replication fork. There it is also a subunit of it. When we deplete CRAC-14, what we know is that those flies are extremely sensitive to DNA damage. Here, those are CRAC-14 mutant flies that are irradiated with gamma irradiation, and they are extremely sick. They have no balance. They cannot hatch. You really have to get them out of the pupae case, etc. What we also know is that when we do a COMET assay, where we basically have single cells and then lyse the cells and have them on an agarose gel and electrophoresis, you can basically see a tail of broken DNA if you treat them with DNA damage agent. And in the CRAC-14 RNAi or mutant cells, you see that tail is over time not repaired. And you can quantify that. Here you see the control. The tails get shorter over time, which means the DNA damage is repaired. That does not happen almost at all in CRAC-14 mutants. So there is a function of DNA damage. We have ectopic CENP-A in those flies. So, you know, the obvious really is CENP-A may actually go to sites of DNA damage. And that's why we see those defects. And that was our model at the time, that you have in wild type, you have DNA damage induction. CRAC-14 is involved in making sure the damage is repaired, but also that CENP-A does not go to those sites of damage, and that normal chromatin is restored. In the absence of CRAC-14, what you have is CENP-A can go to the damaged chromatin. Chromatin is not repaired, and you get genome instability. However, what we did not know at the time, and what we really wanted to find out is where really in the genome does CENP-A go? Where does it incorporate? Does it actually incorporate into sites of DNA damage? And that's when Sarah really took over the project and came up with a really neat but complicating looking analysis to study everything in one cell, basically. So Sarah combined CUT&Tag with an auxin-inducible DNA damage induction system via ACES, which we generously got from the Legube lab called DIVA. And then she did RNAi for CRAC-14 in those cells. So basically, Sarah did CUT&Tag here with CENP-A or HTAV antibodies in cells where she can induce DNA damage by the ACES1 when she washes out auxin so that the ACES is not degraded anymore. And then we did sequencing to see where CENP-A localizes to, or as a control, gamma H2AV. For people who are not working with flies, gamma H2AV is kind of gamma H2AX in human cells that marks DNA damage. Here you can see after washout, 3 or 4 hours after auxin washout, we do see a high induction of gamma H2AV. And so to cut a very long story that required a lot of optimization, very short, is here you see some of the gamma H2AV peak calling that Sarah did to identify ACEs or where the ACEs cutting sites were. And the first problem is that many ACEs sites in the genome, it doesn't always cut at all sites, which can be good or bad. However, we only found 18 common sites in our replicates, and of those 18 common sites, none of them had a coverage of CENP-A. So we really have to conclude from that even though we do have more CENP-A in the genome, and I come to that now, CENP-A does not seem to go through the ACES1 sites of DNA damage, which was our first question. However, we had all the data set and there was still a lot of amazing information in there. So first of all, here those are the replicates I'm going to show. And as you can see here, there is genome-wide, there is more CENP-A in the CRAC-14 depleted cells. And for us, very interestingly, actually, is that most of the, or much of the CENP-A that we covered in the CRAC-14 depletion was actually on multi-mappers. Why is that interesting to us? It's because centromeres are highly repetitive. And here I would really advertise a very nice paper from the La Cruente and Melone lab, where they mapped using long reads and CENP-A chromatin IP, where they actually mapped centromeric islands of mostly transposable elements that are within the satellite repeats where CENP-A nucleosomes localize to. And all of the different chromosomes are slightly different. What they all have in common is that they have a G2-3 element in there or multiple ones. And so you've used their annotation a lot for the mapping here. And so those are basically sequences that are within the multi-mappers. And when we then look specifically at the peaks or the reads at centromeres, we did see that there is more CENP-A in CRAC-14 depletion at centromeres. And here you can see at all the different peaks when we did peak calling, so they all have more CENP-A in here, but you can also see it here, for instance, over this G2-LTR here, we have more CENP-A in the CRAC-14 depletion than in brown. However, also, I come to that later, in the simple repeats that are flanking those centromeric islands, we do see more CENP-A in the CRAC-14 mutant. However, we also looked genome-wide, and we did see that genome-wide also CENP-A goes up. And we look where in the genome we see more CENP-A, and we see it mostly at repetitive regions of simple repeats, retro elements as well, but not at the centromere, at promoters, exons, and at boundary elements. And here that is one example. Here that's the CRAC-14 over an exon here where we have a massive new peak upon CRAC-14 depletion. Here we have one peak at the boundary element, which is also very exciting for the kinetic or centromere field, because in 2011, the Hoyne lab proposed that boundary elements of heterochromatin and euchromatin are actually hotspots for de novo kinetic core formation. And here in those CRAC-14 mutants, we do see an enrichment of CENP-A going specifically there. Also very interesting for us is that when we removed all the centromeric repeats families and looked at the top 20% of other repeats that we found, What was also very high are actually the pericentric satellite repeats. Here, for instance, the 359 base pair repeat belongs to the 1.68A satellite family, where we do see an enrichment when we deplete CRAC-14. And here you can see the coverage, what we have in the CRAC-14 of the 1.688 satellite repeat region. And that is interesting to us because I forgot to mention here the paper, but it's Rogers et al. We showed that actually the RNA of satellite 3, those repeats are transcribed. When you knock it out by LNA, you get massive chromosome segregation defects. So those satellite repeats are also required for chromosome segregation, proper chromosome segregation. So, you know, we really now want to know what is the consequence of this ectopic CENP-A retention in CRAC-14 mutant. And we look at many things, changes in chromatin architecture, but also kinetical formation and chromosome segregation, but also we wanted to know whether there is a change in gene expression when we have more CENP-A in those CRAC-14 mutants. And that's just a summary of the GO analysis when we did RNA-seq in CRAC-14 depleted S2 cells and in embryos. And to our delight, the very top hit is really mitotic cell cycle genes that are expressed in those. It's also DNA damage in there, DNA damage repair, etc. And what was really interesting to us is when we compared our CUT&Tag data with the RNA-seq data, we found some genes where we had an elevated CENP-A level that also are misexpressed. That is here just one example where we have a peak and then also have a misexpression in both directions. Some genes go up when they have CENP-A, other genes are actually downregulated. And so that is very exciting, and that's now what we want to try to understand, whether those misexpressed genes actually lead to the phenotypes that I showed you earlier. However, at the very end, what I wanted to show you, what we are also very excited about, is that in CRAC-14 mutants cells and embryos, also the centromeric repeats are misexpressed. As you can see here in red, those are all centromeric repeats that are upregulated in CRAC-14 mutants. And very interestingly also to us is that here the orange dot that you can see here is actually a telomeric repeat. And why am I now at the last slide starting talking about telomeres is going back to that old paper from Wiener, where we actually were able to see CENP-A localizing to telomeric sites when we actually removed the sheltering complex of the telomeres. So basically, we made a double-strand break at the telomere, and then it wasn't repaired anymore in CRAC-14 mutants, and in this case, we could actually localize CENP-A there. So now our job is to really find the function between misexpressed genes and retro elements and the phenotypes that we see upon CRAC-14 depletion and CENP-A mislocalization. And that brings me to our updated model from what we have now. So CRAC-14 is still required. It's still the same in that model, that CENP-A is loaded correctly, correct amount at the centromere. It's also required for DNA damage repair, CRAC-14. Whether that function has anything to do with CENP-A, we don't know. Actually, we don't think so. However, what we do know is if we remove CRAC-14 from the cell, there is a defect in DNA damage repair. We also get more CENP-A at the centromere. We get more CENP-A at pericentromeres, boundary elements, LTRs, promoters and genes, and at the telomere. And that then causes a change in gene expression, but also segregation and cell cycle defects and misexpression of transposable elements. And that brings me to my summary. So what I showed you here is that non-centromeric CENP-A requires the NERD complex for loading to non-centromeric sites. And I showed you that CRAC-14 is important for DNA damage repair in flies. and that it restricts CENP-A loading to centromeres and genome-wide. But those two things have probably nothing to do with each other. So that's what we think now. CRAC-14 depletion results in additional CENP-A peaks at promoters, genes, repeats, and transposable elements, and higher CENP-A leads to gene expression level changes of genes, centromeric and telomeric repeats. And yeah, the last sentence I already said. With that, I would like to acknowledge the people who have done the work. I introduced Sarah already, she is here, but also the rest of my group. Then other people that we have to thank is the mass spec facility in Heidelberg at the CENP-A, Carsten Rippel's lab, and Carsten for repeat mapping help. Kai Fenzel, he's actually here on the picture, and the Buka lab for sequencing, the Legube lab for the DIVA system, and my funding agency. And in the last 20 seconds, I would like to do a little bit of advertisement. Finally, after 2 years of postponement, hopefully next year from July 24 to 29, we will be able to have an in-person Centromere biology Gordon research conference again that Ian Cheeseman and myself are organizing. Registration just has opened, so please, if you're interested in centromeres, apply. And that's it. If there are any questions, I'm happy to answer questions.
Thank you, Sylvia. That was a really nice talk. I will remind the audience that you can raise your hand and we can unmute you if you want to ask your question yourself, or you can type your questions in the Q&A. Also, if you would like to join the coffee session afterwards to talk to either speaker, please let us know in the chat and we'll be happy to add you to that session. It'll be on the same link. So I don't see any questions yet, but I can start with a couple questions. I was wondering if when you, early on, you showed some work overexpressing CENP-A, and I'm wondering if where it localizes in that overexpression correlates with when you deplete CRAC.
Yeah, I mean, we didn't do it systematically, but in that one slide, I showed you that other lab, Patrick Hoyne's lab, they have overexpressed CENP-A and then looked where it localizes, and they found that a site where it actually stably incorporates. So basically what they did is they gave a pulse of CENP-A overexpression and then sort of let it clear out to some extent. And there were some hotspots, as they called it, where CENP-A maintained. And those were sites where heterochromatin and euchromatin sort of meets boundary elements. And some of those boundary elements, we also do see CENP-A going when we have CRAC-14 depleted. So there is something at those sites that make it more stable when you overexpress it, and that have CENP-A go in our system where we don't overexpress it. So yeah, I really think there is this underlying chromatin structure that you have there helps CENP-A to being maintained there.
Yeah, cool.
Yeah.
The other question I had was from earlier in your talk. I thought it was very interesting how you can delete so much of the N-terminus tail and not see any obvious defects. I'm wondering, are those flies like happy and healthy, like fertility, development, all that is fine?
So what I showed you there is we've done that in cells. However, we also made flies with delta N, and they are mostly fine. So it's a transgene. So they still have the endogenous CENP-A. So that may actually help. I have a little bit of a feedback right now. That may actually help for the cells or the flies to survive. That we don't know. So we would need to deplete the endogenous or cross those transgenic flies into a CENP-A mutant background, which we haven't done, to answer that question correctly.
Yeah, because that would be cool, because I know the N-terminus tail changes a lot. Like that's something that we study in the lab here, is how there's so much difference in the N-terminus tail, even the histone fold's pretty conserved.
Yeah. We didn't expect that either, that you can basically remove the whole thing. Yeah. And we know from the work that we've done with the NERD complex that some components of the NERD complex actually interact with the N-terminal tail as well. Yeah.
Cool. Let me